Working with eukaryotes can be pretty complicated, even more so when you're working with something like Cryptococcus. Cryptococcus is an encapsulated yeast, which means that it's surrounded by a thick, goopy envelope rich in polysaccharides. This capsule is pretty important in understanding virulence because it induces inflammation and inhibits phagocytosis by alveolar macrophages. But when it comes to understanding Cryptococcus, its capsule can sometimes get in the way. Usually when we're trying to get a snapshot of the cryptococcal genome or proteome, we chew away the cell wall and capsule with a special type of cellulase that breaks down the cell into a fragile, cell wall-less ball called a protoplast. First thing we're going to do is take a couple of loopfuls of Cryptococcus gattii that we've had growing at 30 degrees Celsius for a couple of days. We're going to suspend these mature cells in a buffer called SCE, sorbitol, citrate, and EDTA. The high molarity of sorbitol in this buffer acts as a sort of osmotic support, providing a gentler, isotonic environment so that the sensitive protoplasts maintain their integrity. Once the cells are resuspended in the SCE, we're going to give them a quick spin down to collect them into a pellet. We're then going to rinse that pellet in SCE, making sure the cells are nice and clean before we start chewing them up. Finally, we resuspend in some SCE that we've dissolved some of our gluconex cellulase in. We then activate and incubate our cell enzyme suspension at 37 degrees Celsius for 85 minutes. It's vital that we don't incubate our cells for too long, as the cellulase can continue to break down the polysaccharides until they're too small for us to easily remove from our DNA suspension. Once that's done, we can keep our protoplasts cool by leaving them on ice. This deactivates the enzyme, making sure our protoplasts are ready to start extracting stuff out of them.